Hello, we are the Biological Anthropology Group, and the title of our project is Biodiversity and Conservation, which we undertook with the Prime Research Group. My name is Vishala.
My name is Saki Kanai.
My name is Marina Sato.
My name is Sophie.
My name is Sakiho Tanaka.
My name is Kiriko.
My name is Shiori Okamoto.
PRIME stands for Primate Immunogenetics and Molecular Ecology. It is the study of genetic variation and evolutionary biology in humans and primates. Research is also conducted on the genetic diversity in other mammals and endangered species in Central and South America, such as Darwin's fox.
The danger of variety of life between or within a species, ecocycle, or environment on Earth. The components biodiversity are genetic diversity, genetic variation within population of species, species diversity, number of species in an area, diversity in higher taxa, variation between genera, family, order, etc. Conservation. Wildlife conservation has become an increasingly important practice due to the negative effects of human activity on biodiversity. Biodiversity can be measured by assessing the global conservation status of certain species. The world's most comprehensive inventory of the global conservation status of biological species.
Okay, so a big part of our project was hearing about the research undertaken by other people actually currently working out in the field. An example of which was Dr. Kat Coops, whose work centres on chimpanzees in the Nimba Mountains, Guinea, Africa. This is an example of in situ conservation. The subjects are studied in their natural environments, and the camp at this particular site has existed all year round for a total of 9 years. This means that the chimps have come to regard humans as non-threats because their presence is almost constant, and therefore behave naturally around them. This makes behavioural observations more reliable. Sources of DNA extracted in order to study this particular group of chimpanzees could be extracted in a variety of ways, four of which are listed above. These are examples of non-invasive techniques as they do not disturb the animal's natural routines. These are used in preference to invasive techniques, which are samples of blood or tissue samples, as they require you to catch, sedate, and then re-release the animal, which is obviously not in their best interests.
We also heard from Professor Antonio Mendez Pontes about endangered primates in South America. These are 7 different species of primate found in South America, Amazon, Brazil. Their status vary from threatened to endangered. I will talk you more in detail about one in particular. This is Cebus monkey, and their color is light brown to mustard yellow to black, and the tail is long and prehensile. They eat vegetables, seeds, and insects. They have polygamous mating system, and they may live to 45 years old. Their conservation status is certain by hunting and habitat destruction.
I will explain the Ethogram. Ethology, a catalog or inventory of all behaviors in an animal. For example, looking around and grooming others. This is an example of an Ethogram and the type of observation we want to make.
First, divide the field exactly. Its interval is 5 kilometers and go backwards and forwards on this line. Next, be careful of angles, right and left, also vertical. We used standard methods, feature animals, and scan. We can observe species, diet, drinking, eating, interspecific, interactions, sexual behavior, and daily actions. Our project also involved exchange conversation.
The Darwin fox was the subject's food fix we used for the DNA extraction. First documented by Charles Darwin. DNA extraction, PCR, and Gale electrophoresis. We extracted DNA from fecal samples of the Darwin fox on first day. On second day, we used this sample of PCR polymerase chain reaction to amplify sections of mitochondrial DNA, cytochrome B, and the D loop. We chose these sections as they mutate rapidly and change accumulate fast enough for identification of individuals within a species and between species.
Following the PCR, we tested the success of our experiments using gel electrophoresis. The cytochrome B PCR was more successful than our D loop, as unfortunately a negative control, seen far left, was contaminated. The brighter each band, the more DNA present. In the case of the control, there should be none.
Okay, we had 5 successful DNA samples, 2 which turned out to be the foxes we were looking for, 2 dogs, and 1 rare Chilean bobcat, which actually is a species not yet described.
We visited Linton Zoo, where we carried out our ex situ observations and ethograms. We studied 2 types of lemurs, and in particular the crowned lemur. The male and female lemurs are very different. The females are grey with a pale orange crown, while the males are a darker reddish brown, crowned with black and orange. It was important to clearly distinguish between the lemurs in order to ensure that the data was recorded for the respective lemurs on the ethogram. Recently, lemurs have been exploited for bushmeat, which has led to the species becoming vulnerable. Here are a few of our observations at Linton Zoo. In this picture, you can see that there's a red ruffed lemur and it's been circled below. This is an example of interspecific interaction between lemurs, where the crowned lemur is seen to be more alert and marks its territory as it's being intimidated by the red ruffed lemur. We also got some videos of some activity of the lemurs. This video shows one of the crowned lemurs scent marking, and this is very typical in its natural habitat. And we were quite surprised by this because it's in a zoo, so we were happy that it exhibited normal behaviour. We also studied the snow leopard, which is currently also an endangered species. Here is our ethogram of the snow leopard and other observations. So we took an ethogram of the snow leopard and we timed it, we took it at different time intervals, and here are our observations. So you'll just see the snow leopard pacing around and it's swatting flies with its paw and rolling around in the sun, as normal leopards do in the wild.
What we have learned: how to make behavioral observations, the importance of conservation, the use of genetics in furthering our understanding of animals in situ and ex situ.
And we'd just like to say a really big thank you to Dr. Knapp, Becky, Jan, Nozomi, and the researchers that came to talk to us for making it such a unique experience. And thank you for watching.